Hello and welcome to Histology 101. I'm Jamie Chapman and today we're going to be having a look at the stomach and the intestines. So in our first ever episode we looked at the oral cavity, so we looked at the tongue and we looked at the lip and we looked at the salivary glands, and hopefully you learned a little bit about that. And so today we're going to continue our look at the gastrointestinal system. We're going to look at make our way down the rest of the gastrointestinal tract. So we're going to have a look at the junction between the esophagus and the stomach and the things to look out for there. And then we're going to move into the stomach itself, look at the different regions, and then, if time permits, have a look at the different regions of the intestines, finishing at the ano-rectal canal. That's a lot to get in. We'll see how much we can get through. What we can't get through today, we'll obviously try to revisit at another future stream. Please come and say g'day in the chat so I know I'm not talking to myself. Once again, Dr. James Crane is here next to me. I am wearing headphones this time. I'm hoping to hear any announcements and things. So first of all, Isabella HS99 followed about 8 minutes ago, so thank you very much for the follow, Isabella. Great to have you along if you're in there. James has just said hello in the chat room there. So how about we get started? All right, so what we're looking at here is a section of the junction between the esophagus and the stomach. So on the right hand side here we actually have the esophagus, and then we've got the first part of the stomach, which is known as the cardiac region of the stomach, and then we have the body of the stomach continuing on this way. Now, if you remember from last week, those of you who were here, the esophagus and the most of the rest of the gastrointestinal tract is made up of 4 layers. It's made up of a mucosa consisting of an epithelium, underlying layer of loose connective tissue called the lamina propria. Then we have a layer of smooth muscle here called the muscularis mucosae. Then we have another layer of dense irregular connective tissue known as the submucosa, so tunica mucosa, tunica submucosa. Then we have the tunica muscularis, which is usually made up of an inner circular outer longitudinal layer that varies along the tract, along the way, and then either a tunica adventitia or a tunica serosa. And either that's a simple mesothelium. I think we zoom in here, we can actually see some of these mesothelial cells on the surface here. So this is a mesothelium here. So this is a tunica serosa. If it was an adventitia, then this would just be connective tissue and you wouldn't have this, this lining here, this simple squamous lining. All right, so the 4 layers, and if we remember, the esophagus is made up of a stratified squamous non-keratinized epithelium. So we can see that here. It's got a thick stratified epithelium, nuclei all the way to the surface there. So this is a stratified squamous non-keratinized layer. Here's our lamina propria, and then here's our muscularis mucosa. So if we continue along the esophagus, we can see these folds, typical of an organ that has a well-developed muscularis layer. And then we'll see a junction between the esophagus and the stomach, where we have this quite dramatic transformation of the epithelial surface from a stratified squamous epithelium to a simple columnar epithelium, and here it is here. So if we zoom in, so here's our stratified squamous non-keratinized epithelium, and here we've got these layer of simple columnar cells. These are surface mucus cells, and they produce a protective mucus. It lines the surface, and of course that protects this region from acid and enzymes. So these are surface mucus cells. They're not goblet cells. We'll see goblet cells in the gastrointestinal tract, but we won't actually see them until we reach the intestines. So the stomach does produce lots of mucus. It just doesn't come from goblet cells like we'll see a bit later. So that one of the sites of the mucus is these surface mucus cells. Now you'll see these sort of little depressions here, these little depressions in the top part of this mucosa here. These are known as the gastric pits, and you can actually use the depth of these pits as a good guide to which of the 3 regions of the stomach we're looking at. So histologically, we recognize 3 regions of the stomach. The first region, which is this small region at the junction here, this is known as the cardiac region of the stomach, and it's characterized by having these gastric pits, which are about 40% of the depth of the entire mucosa, and they're also characterized by having these mucus secreting glands, and that's what these are here. So these are known as cardiac gastric glands, or just cardiac glands, and they're mucus secreting. You can see a very pale staining apical cytoplasm here. And the other feature that it has a lot of is lamina propria in between those glands. So they're quite sparse glands. They're not very densely packed together. They're quite sparse. There's a lot of loose connective tissue there in between the glands. And it's very characteristic of this region of the stomach, the cardiac region of the stomach. The second region of the stomach, which we can actually see if we move along a little bit further, is known as the body, or the fundus, or body and fundus, histologically identical together. And this is what it looks like here. It's characterized by having these different cell types within the glands. The top of the cells predominantly have these very acidophilic cells that have these lovely round nuclei, often described as having sort of a fried egg type of appearance. So this would be the egg white, and the egg yolk is the round nucleus in the center here. These cells are known as parietal cells, and they're responsible for the secretion of hydrogen chloride ions, which eventually fuses together to form hydrochloric acid in the lumen of the stomach. Now, the reason they're so acidophilic is that they have lots of intramembranous foldings, sort of like intramembranous microvilli almost. They actually use those membranes to have lots of ion channels to pump out lots of hydrogen ions and lots of chloride ions. And they also have lots of mitochondria to help power that production of those ions and release of those ions. So together, the intramembranous folding, so lots of membranes means lots of acidophilia, and lots of mitochondria, which are also very acidophilic, leads to the very acidophilic nature of, or staining pattern of, these parietal cells. Now, the parietal cells actually start off have a predominant location near the top of the gland. They're so predominantly found at the top of the gland, and that often gives the top of the glands of the body and fundus regions of the stomach a very acidophilic staining pattern. The base of the glands, you can see here, look a bit more purple, a bit more basophilic, and that's because they predominantly contain a different cell type known as the chief cells. Now, the chief cells are responsible for the secretion, or the production and secretion of pepsinogen, which is a protease. It's an inactive protease that becomes activated once it actually reaches the lumen and comes into contact with the hydrochloric acid. So that way, the stomach doesn't begin to auto-digest itself. So it releases it as an inactive form in the form of pepsinogen, which is then cleaved to form the active pepsin in the presence of the hydrochloric acid in the stomach. So these are chief cells, and as I say, they're predominantly localized to the base of the gastric glands in the body fundus region of the stomach. Not shown here is the third region of the stomach, which we'll look at in another slide, but that's the pyloric region of the stomach, and that's characterized by having very deep gastric pits. Now, I didn't talk about the pits for the body region because we're actually going to look at a separate slide of the body of the stomach to point out these features again, but I just wanted to point out the two regions, and then, as I say, the third region is the pyloric region of the stomach, and we'll look at that in a little while. And the pyloric region, of course, is the part that joins on to the duodenum. So quite a dramatic change in epithelium. So if you're looking for a junction, you're looking for a change in epithelium from this stratified squamous non-keratinized to a simple columnar. There's no true sort of muscular sphincter here, so there's no real thickening of the muscularis layer like we see in some of the other regions, like the pyloric sphincter, for example. There's a thickening of the inner circular layer of the muscularis, whereas we don't see that here in the lower esophageal sphincter, or with the junction with the cardiac region. So that's the main sort of features that I really wanted to point out here. Everything else is pretty much the same. During, as we move along to the stomach, it does gain a third layer of muscularis, an oblique innermost layer of muscularis, which is unique to the stomach, or have actually found that that's quite tricky to identify in these sections. So does anyone have any questions about the cardio-oesophageal junction before we move on? So what you might see here is a little bit of a mucosa-associated lymphoid tissue, so a little bit of lymphocyte aggregation here, sampling the material that's coming through here. You can see this little accumulation of lymphocytes here. But otherwise, what you look for is gastric pits, which are about 40% the depth, very sparse mucus-secreting glands, so there's no evidence of parietal cells or anything else. These are actually made up of mucous neck cells and enteroendocrinocytes. And the enteroendocrinocytes are a bit tricky to see. I don't think I've ever seen them actually in any of these slides here, but sometimes you can get some really nice sections and actually see the enteroendocrine cells, particularly at the base of the glands. All right, so it's very quiet in the chat room, so I assume that there's no questions. So we'll move on to the next slide, which is actually looking again at the body region of the stomach. Okay, so this is a section of the body region of the stomach. As I mentioned before, the stomach histologically is divided into those three regions, and we actually group together the body and the fundus of the stomach because histologically they're identical. So we call them the body fundus region of the stomach. You can see it's a very glandular type of lining. Almost all of this is glands. If we sort of zoom in, I actually like this part here, so I'm just going to flip it around. Don't get seasick. Okay, so if you have a look here, it's actually got some mucus on the surface here. So these are being secreted by these surface mucus cells, and you can see the gastric pits are very, very shallow, just these little indents here. Around about 10 to 20% of the depth of the mucosa is the depth of the gastric pits of the body fundus region of the stomach. And then all of this is glands, and it's so tightly packed together. It's just filled with glands, and you can barely see any lamina propria between the glands, even though there is some. So if you sort of zoom in a little bit, you might be able to see some of these sort of flattened cells in between, and these are collagen fibers here. So this is part of the lamina propria between all of these glands. So this is all glandular cell out here. So these are the gastric glands, and they're emptying into these gastric pits. They're classified as sort of simple branch coiled tubular glands. And you can see that the top of the glands quite pink and the bottom of the glands quite purple, and again that's a reflection of the distribution of cell types that we find within these glands here. So lots of parietal cells in the top of the glands and lots of chief cells down the bottom. There are two other cell types which we find in these glands of the body fundus. Those are the mucous neck cells. They're not always that easy to see, to be honest with you, unless you use a special type of stain specifically for mucin. Yeah, it's a bit tricky. Maybe something like that might be a mucous neck cell. You can see the sort of paler cytoplasms here. These might be mucous neck cells. So that's the third cell type. You've got your parietal cells, the very acidophilic cells with the nice round nucleus. You've got your mucous neck cells. You've got your chief cells, and then also, like we were describing before, you can have enteroendocrine cells, but as I say, they're a bit trickier to identify, so we certainly wouldn't expect for our medical students anyway, to expect them to be able to identify them. So again, yeah, we've got that lovely distribution of the parietal cells at the top here, very acidophilic, because of the reasons I mentioned before. We've got these cells down here. Now, typically, what we would expect to see of a chief cell is basal basophilia related to their accumulation of rough endoplasmic reticulum, and then apical acidophilia related to the storage of those zymogen granules that are containing the pepsinogen that attracts the eosin stain. But it's likely that whichever animal that this was taken from had a meal relatively recently, and probably a lot of these granules have been exocytosed. So they're relatively pale staining, but it does give a general overview of basophilia. MJ Green just asked, hey Jamie, are G cells enteroendocrine cells? Yeah, so that's just another name that we give to the enteroendocrine cells of the stomach. They're called G cells because they secrete gastrin. and that's a hormone which is released into the bloodstream and stimulates the secretion of hydrochloric acid and the pepsinogen. So yeah, so G cells are definitely a part of the enteroendocrine cells. So when we get to the small intestine, you can have cells which secrete CCK and cholecystokinin, and secretin, and they're often named accordingly as well. So S cells for secretin cells and so on. Yeah, so no worries. The classic sort of feature of it, very shallow gastric pits, lots and lots of glands, just so packed. You really can't confuse the body fundus of the stomach for any other region of the stomach, I don't think. Very little evidence of lamina propria in between the glands. And then you've got this sort of staining differentiation of the acidophilic nature to the top of the glands because of the higher concentration of parietal cells, and then the very basophilic nature of the basal region because of the higher concentration of chief cells. Here we have the muscularis mucosae. This is probably not of interest to you, but I think it's kind of cool. You can actually see that the muscularis mucosae is actually orientated here in different directions. So it's not really described in textbooks. Often it's described as having two layers, an inner circular, outer longitudinal layer. But here you can clearly see that there's three orientations. So that's a bit of a unique finding, which I think is kind of cool. Yeah, so then we move into the submucosa. You can see some blood vessels out here, dense irregular connective tissue. You can see some adipocytes. So these are some adipocytes out here. There's a little blood vessel, little arteriole or something here. Some more adipocytes. Can't really see any evidence of, any of the plexuses related to the myenteric plexus. Sometimes it takes a little while to see those sort of things. But I can't really see any evidence of them there. You can still, again, see some more blood vessels out here. Then we have our muscularis layer, inner circular and outer longitudinal layer. This is actually part of the myenteric plexus here. So we can actually see this big, I don't know how well you can see that. So it's actually not very well focused in this slide scan, but this pale nucleus here, the prominent nucleolus, very classic of a neuronal nucleus of a soma. And so this is actually probably one of these ganglion cells related to the enteric nervous system. And so if you have a look at that, if you look between the two layers of muscle, you can often find these accumulations of neurons related to that as well. There's a nice one there as well. See this big pale nucleus and prominent nucleolus. So that's a neuron, a soma there. And these are probably some other ones over here as well. Okay, so that's the, out here you've got some nice adipose tissue, some larger blood vessels and so on out here. Does anyone have any questions about the body fundus of the stomach before we move on? I think it's kind of cool, even at this low mag, you can see that difference in staining between the two regions there. Gives this nice banding type of structure. You can see lots of these surface mucus cells along the top here, very pale staining, all the nuclei in a row. Again, these are not goblet cells, but surface mucus cells. All right, we're all quiet in the chat again. So let's move to our next region. Is it tunica serosa in the stomach? Yeah, it is a tunica serosa in the stomach. The stomach isn't fixed because the stomach really needs to be able to churn. So the fact that it can churn and move around and mix the food contents with the secretions of the stomach, the gastric juices, that means it's mobile and has a potential for abrasion. So any organ that actually requires sort of mobility to enable mixing and that sort of stuff will have a serosa on the outside because it provides a smooth, frictionless surface on the outer surface, and so it doesn't cause abrasion. So I think we can zoom out here a little bit. You can actually see some of the simple squamous mesothelial lining of the serosa, tunica serosa. No problem, Hibiscus. Thank you for asking the question. I'm really glad people are interacting, because that's what we really want for this. We want you guys to have a real-time interaction and be able to ask questions and get things clarified. Some really nice serosa out here again. You can see the large amount of adipose, but the serosa on the outside there. All right, well, if there's no more questions, we'll move on to the next slide, which is the pyloric region of the stomach. Now, this actually has some nice folds. Does anyone remember what the folds in the stomach are called? Let's see if we can get some answers in the chat here. Rugae, yes, Hibiscus, well done. Yeah, so they're called rugae. So this is a ruga, one of them. So you can see numerous rugae here. Izzy Rob, well done to support the people contributing in the chat. It's really great. Rugae, Histo 101, yes, correct, well done. So rugae are folds of both the mucosa, which is this layer here, and the submucosa, which is this layer here. Now, these are non-permanent folds, so if there was food moving through here, this could expand and they would flatten out. And then when it shrinks up again, you get these rugae appearing once again. Now, I really like this slide. I think it's really well stained and shows some of these features of the pyloric region of the stomach, being classically that we have these very deep gastric pits. So you can see this lumen here. I might just rotate it a little bit so you can see it. So these are the deep gastric pits, and then the glands here are mucus secreting, so entirely mucus secreting glands, flattened basally located nuclei, very pale staining apical cytoplasms, and you can see a relative amount of lamina propria once again between these glands and between the gastric pits here. So certainly you couldn't confuse this for the body fundus region of the stomach. James has just found the definition of rugae. Ruga is the singular form, rugae being the plural form. So here's the lamina propria, and here's our glands from the pyloric region of the stomach. Now, these are made up of mucous neck cells, and there would be enteroendocrine cells here once again. And again, they are pretty tricky to see, those enteroendocrine cells. So you wouldn't be expected to identify them, certainly not in my class. So here we have the muscularis mucosae. Again, we can see a couple of layers here for the muscularis mucosae, and then we've got our serosa. So the classic features of the pyloric region of the stomach are the very deep gastric pits, the mucus secreting pyloric glands of the stomach, which are entirely mucus secreting. And those are the main features. We will see, if we were to have a look, well, we will look at the junction between the pyloric and the duodenum. We will see evidence of the pyloric sphincter, which is a thickening of the inner circular layer, so it's a true muscular sphincter there. So what you look for if you're trying to identify whether you're looking at the stomach, which region of the stomach, look at the depth of the gastric pits, look at the cellular composition of the glands. Those are the main ways to distinguish between the types. Again, pyloric region, very deep gastric pits, about 50% or so of the depth of the mucosa, and entirely mucus secreting. The lamina propria will help guide you whether you're looking at cardiac / pylorus versus the body of the stomach. You almost never see lamina propria in the body fundus region of the stomach. These are lovely sections. I think they're really, it's almost like someone's drawn them. They almost look cartoon-like. I don't know. That's just me. You guys know I'm a bit mental about histology. So we've got some blood vessels in the submucosa here. We've got our muscularis layer on the outside. Those are the main features there of the pyloric region of the stomach. Now, the next slide we're going to look at is one which students had a little bit of difficulty interpreting, and it is a very complicated slide because it's unfortunately folded several times. So you've got some mucosa here, and then you've got some mucosa here, which really doesn't make any sense. But unfortunately, when we processed it, this bit of tissue actually folded upon itself. But the actual junction between the pyloric region of the stomach and the duodenum is this region here. You can sort of see this change just even at this low magnification from this flat surface to these finger-like projections of the villi of the duodenum there. You can also see this thickening of this. So here's the normal thickness of the inner circular layer of the tunica muscularis, and then you've got this big thick region here. This is the pyloric sphincter. So this is helping to regulate the exit of chyme, acidic chyme, into the duodenum, so that it's not overwhelmed by all of this acid and enzymes from the stomach. The other feature to look out for, to look at whether you're looking at a section of the one of the junctions, or which junction you're looking at, is the presence of submucosal glands. Now, we can have submucosal glands in the esophagus, and that they can be found in the distal region near the junction between the esophagus and the stomach, the cardio-oesophageal junction. But there we're moving from an epithelium that is stratified squamous, non-carcinized, to a simple columnar, whereas here we're actually moving from a simple columnar to a simple columnar. You won't see that epithelial transition, with the exception that we're going to have different cell types. You know, we're actually going to see evidence of goblet cells for the first time in our track down the gastrointestinal tract. So these structures here, they've come a little bit early. So if we think about the junction being around about this region here in terms of the epithelial change or the mucosal change, we can actually see these glands down here within the submucosa. These are the Brunner's glands. These are the mucus secreting glands that provide a protective mucus lining for the duodenum here from the acid secretions of the acidic chyme. So they can sometimes start a little bit early. You can see they're still in the stomach, the pyloric region of the stomach here. Again, we've got these mucus secreting glands here, and in fact these look very, very similar to the Brunner's glands, which we'll see in a little while. Basally located flattened nuclei, very pale staining cytoplasm here. These are mucus secreting glands. And then unfortunately the epithelium's a bit disrupted here. So if we keep moving down, again we can actually see here's the muscularis mucosa, and then so this must be the submucosa, and you can see histologically these Brunner's glands look very similar to the pyloric glands here. So you can see them in the submucosa here. A bit further down we've got some of these glands as well. So these are Brunner's glands, which are part of the small intestine, the duodenum in particular. They've come a little bit early and still in the stomach here. So nice glands here, nice lumen present here, and then you can see them again, once again, further down in the submucosa. And we often see their ducts. You can see them sort of emptying into the base of the gastric pits here, and then eventually these will empty into the base of the intestinal glands. Again, you can see that in this region the muscularis mucosa is quite disrupted in this region, because we've got so many of these glands sort of making their way through. You can see this nice region here where you've got the muscularis mucosa and these Brunner's glands emptying into the base of this region here. So again, we're still stomach here. This is still surface mucus cells. These are the gastric pits. So these are the mucus secreting glands, and now we begin to see these little round structures, these little circles. These are actually goblet cells. So we begin to see evidence of goblet cells for the first time, which we didn't see before in any of the stomach sections. And we've got these finger-like projections. We've got our first evidence of villi. And so these are our enterocytes, which have this very well-developed brush border, very acidophilic, and then this core of lamina propria. So these are our villi here, and then these are our intestinal glands, or crypts of Lieberkühn, as they're known. And you can see here, this is really lovely, this one. You can see that these Brunner's glands are emptying into the base of one of these crypts of Lieberkühn, or the intestinal glands here. So quite a transition from the intestinal glands to the duct cells of these mucus secreting glands. Again, we've got the disrupted muscularis mucosae here. So What you look for the Pyloro-duodenal junction, or the junction between the stomach and the duodenum, is the first evidence of the villi. Look for the presence of Brunner's glands, and if you can have a look low mag, look for the thickening of the inner circular layer of the tunica muscularis or the pyloric sphincter. Those are the major things to be looking out for. So again, that we're moving from a simple columnar to a simple columnar epithelium, but we're not moving like we saw with the cardio-oesophageal region, and we'll begin to see goblet cells in the mucosa there. Pyloric duodenal junction is simple columnar to simple columnar, while cardio-oesophageal stratified squamous to simple columnar. Yes. Both would have submucosal glands, yes. Although that section that I've showed you of the cardio-oesophageal junction never actually had any submucosal glands. So it's mentioned in the textbook, but I didn't actually see any in the slide, and to be honest, I've never actually really seen any evidence of submucosal glands in these sections. So there's no glands, submucosal glands here, and I certainly don't own a slide with any of the glands present. But yes, potentially they both, Histo 101, they both potentially could have submucosal glands. And they're the two only sites in the gastrointestinal tract that can have submucosal glands, so the esophagus, proximal distal regions, and the duodenum. All right, so that was the junction between the pyloric region and the duodenum. We can now have a look at the small intestine, and the students at UTAS, medical students at UTAS, will be very familiar with this slide. I think we've looked at this around about 1 billion times. So this is like the first slide we ever look at in histology. We look at the sort of staining patterns related to understanding about basophilia and acidophilia and so on. No worries, Histo 101. Thanks for the question and clarification. So this is the small intestine. If we zoom in here, it's characterized by having these finger-like projections, which are the villi. It sort of means that these can actually increase surface area for absorption. If we look at their structure in a little bit more detail, we can see they're made up predominantly of these two cell types. These simple columnar cells with the very prominent brush border. These are known as the enterocytes, and they're responsible for the digestion and absorption of nutrients. Now, this brush border are actually microvilli, and on the surface of the microvilli we can have enzymes attached to them, which continue the breakdown of products that have begun to be broken down by secretions from the pancreas. So we have oligosaccharidases and disaccharidases, which break down carbohydrates into their smaller monosaccharide building blocks that can be absorbed by these cells. We have peptidases and things like that can all do that. We also have a really helpful enzyme called enterokinase, and that actually is responsible for the activation of one of the secretions from the pancreas known as trypsinogen, and it activates that to trypsin, and then trypsin goes on and activates all the other inactive enzymes, so it's a really helpful thing. The second cell type, which is very prominent here, are the goblet cells, and you can see them here. These are some really actually lovely sections of goblet cells. You can see the nucleus of the goblet cell and the apical pale staining region of the goblet cell here. So these goblet cells have 1 to 2 secretory cycles during their lifetime. So they actually undergo what's known as compound exocytosis. So all of their secretory organelles sort of fuse together in one big clump and then just sort of exude onto the surface. Onto the surface there. So you can see those as these sort of pale regions scattered throughout. These are really nice here. Then we have the underlying layer of lamina propria with various cell types within here. As a well-developed, robust, innate immune system present here. So we'll find macrophages. You can see evidence of neutrophils out here as well, smooth muscle cells, endothelial cells. The odd plasma cell will be found here too. So what we're looking for a plasma cell is a basophilic cytoplasm. So I'm sure if we zoom around, it's not particularly important to be able to identify these at all, but if you have a look around, you can sometimes see these basophilic cytoplasms, and that's because they have lots of rough endoplasmic reticulum. So that's the villi. They're the cells which line the villi originate from the base of these intestinal glands, or these crypts of Lieberkühn. So often down the base of these glands we can see these little very dense accumulations of chromosomes. These are actually mitotic figures. So these cells are undergoing mitosis and then they migrate their way up. So we're forming new goblet cells. You can see some goblet cells down here. In this region we can sometimes, certainly they're not very obvious in this section, but in other sections you might be able to spot a different cell type known as Paneth cells. So Paneth cells are often found down the base of the intestinal glands, and they are one of the innate immune cells. They secrete things like phospholipase A2 and lysozyme and other substances. They often have very acidophilic apical granules. They're not very evident in this section here, so I'm sorry about that. But again, you can see these mitotic figures. And the other cell type we can often see are enteroendocrine cells. So that's the other cell type, and stem cells, which are the mitotic figures here. So lots of these intestinal glands here. They're classified as simple coiled tubular glands, so it's very rare to actually get a gland all the way from the bottom all the way to the top where you can see the lumen, and that's actually helpful because we can use the shape of the glands as a way to identify the large intestine, which has simple straight tubular glands. Some nice evidence of some glands down here. You can see some of these, wait for it to catch up, mitotic figures happening here. Yeah. So then we have, of course, the submucosa. Like we have in the rest of the gastrointestinal tract, we have an inner circular outer longitudinal layer. Sorry, this is a bit folded, this region here. So here's the inner circular layer, outer longitudinal layer, and then in this region we have a serosa. So we can zoom in here. You can see this simple squamous nuclei on the surface. So there's a mesothelium here. So this would be, this part of the small intestine has a serosa. Oops. Yeah. So those are the main features here. I forgot to mention the muscularis mucosae. That's just here. So here's our lamina propria. Here's our muscularis mucosae. So all of these components of the mucosa. Here we have the submucosa, and then we've got the muscularis. You can see some of these cells here. This is part of, James is going to be able to help me here because I can never remember myenteric versus Meissner's. Which one's in the submucosa and which one's, Google it, dude. James is going to Google it.
You looked it up, right?
Sorry?
You did it at the prac.
Yeah, I know, and we've forgotten it already. So the enteric nervous system has those sort of two subdivisions. One's in the submucosa, and then the other one's between the two layers. So here's some of these neurons between the two layers here. And they have names. And I apologise that I don't know the names off the top of my head. I always get them mixed up. One is Meissner's and one is Auerbach's. I might think... does anyone in chat remember where Auerbach's versus Meissner's plexus are located?
Meissner's plexus is submucosal.
Right, so these are the Meissner's plexus. You can see the big pale staining nuclei, prominent nucleolus here. So this is the Meissner's plexus, and then the Auerbach is the one that's found between the two layers. So you can see the big nuclei here, pale staining nucleus, prominent nucleolus. All right, any questions about that? Rip. Got a little bit of pancreas over here. This one, for some reason, doesn't have a lot of evidence of islets or Langhans for some reason, and I'm not entirely sure why. All right, so next slide. Right, so this is a special stain. So we've actually used a stain called Alcian blue here. Thanks for posting that, James. where Alcian blue stains mucus this sort of blue colour, and it's really lovely actually. If we zoom in here, you can see the distribution of the goblet cells by these blue regions. So these are all the goblet cells. So you can actually see them make their way originating from the base of these intestinal glands all the way to the surface of these villi here. And then you've actually got some of this sort of mucus secreting on the surface here, and this looks like some plant material. This regular pattern here, I think this is plant material that this animal had eaten there. This also has well-developed, this region here has well-developed lymphoid tissue, so we've got MALT present here in that region as well. So you can see here a little bit the red here is collagen, so this is actually part of the submucosa, and then we've got the lamina propria. And you can see there's not a lot of fibres in the lamina propria. It's a very loose connective tissue. I was hoping to see if I could see some Pannis cells, but I can't see any in this section. So we'll just move to the next slide. Right, so this is the ileum, and these are the Peyer's patches, which are very classic of the ileum. These are permanent aggregations of mucosa-associated lymphoid tissue. You can see it's very well developed. It's found in the submucosa, and it extends into the mucosa here, and in fact it disrupts some of these villi. So many lymphocytes, so basophilic present here. Very disrupted looking villus here, and then that extends down into this region here. So this is MALT, and these are the Peyer's patches. I don't think, if we zoom in, no, I can't see any. I keep looking, keeping an eye out for Pannis cells. Unfortunately, I can't see them there. Here's our muscularis mucosa, here's our submucosa, and we've got our layers again on the outside. So I just wanted to point out the ileum, which has these permanent aggregations of MALT, which is the Peyer's patches. You can see some mucus secretion here, trapping particles and things in the small intestine there. So again, their villi look very similar to the other villi. I mean, histologically, they say you can distinguish between the different regions, but I certainly don't expect you to be able to do that. It depends on who's teaching you histology, I suppose. So from the small intestine, we then move into the large intestine. Now, I don't have a section of the ileocolic junction. There is a valve there which regulates entry into the large intestine. Students of mine, students doing CAM 202 or students doing CAM 102 that will eventually become 202 students, don't ever expect to see an ileocolic junction in my exams. So the large intestine, now we can tell this is actually not taken from a human. The reason that we can tell it's not from a human is because it actually has a complete outer longitudinal layer. In humans, the outer longitudinal layer is actually arranged into 3 bands known as tineocoli, and that's not present here. So normally, if this was human, we see one large band and then a very, very thin layer of outer longitudinal muscle. But here we've got one thick continuous layer, so that moves us away from actually looking at a section of the human large intestine. Now, how can we tell that this is the large intestine? Well, if we have a look here, have a look at this. We can actually see the lumen from the bottom all the way to the top. This is a simple, straight tubular gland. This is the only location in the GIT that has a simple, straight tubular gland. You'll never see the lumen all the way from the top to the bottom in the rest of the GIT. All the others are coiled. But the large intestine has these simple, straight tubular glands. You can see here that the goblet cells are actually producing quite a basophilic looking mucus. So there must be lots of acids on the surface, sialic acid on the surface of the glycoprotein that makes up mucus here, which is accounting for the acidophilia. So we don't have those surface mucosal projections of villi like we saw in the small intestine. We've got a flat surface, which can sometimes cause students to misidentify this as the stomach. Of course, the difference being, look at all these round structures of the goblet cells. If you see these round structures, then you can't be in the stomach because there's no goblet cells in the stomach. So lots of cross sections of these glands. Again, we get these mitotic figures where the base of the cells, these cells are undergoing mitosis, and they're either forming enterocytes, which are absorbing water, or goblet cells, or enteroendocrine cells. Those are the main cell types that we find in the large intestine glands. Here again, we've got a muscularis mucosae, we've got our submucosa, we've got our muscularis intercircular outer longitudinal, and this region has a serosa. We've got this flattened surface here. Sometimes we can have these accumulations of malt. You can see a little accumulation here, and there's another one along the length over here as well. You can see that's mucosal based, just sort of peeking a little bit into the submucosa there. Lots of these blood vessels here in the submucosa. All right, so I think we've got some more sections here. Again, we've got the large intestine. How can we tell? We've got glands that you can see the lumen all the way from the top to the bottom. Okay, again, we've got this big accumulation of malt here, very basophilic. This pale region here indicates that it's been activated. So normally you can have what's known as a primary follicle or a secondary follicle. So primary follicles don't have this pale center. This pale center here is known as a germinal center, indicating that the cells here are being stimulated and activated, and the B cells are transforming into plasma cells. So they're getting more cytoplasm, therefore they're more separated, and it appears a little bit paler in appearance. Again, if we zoom in, you can see lots of these goblet cells, lots of enterocytes, and they're responsible for absorbing water. Again, the intestinal glands, lots of mitotic figures down the base there. Once again, this is not a human section because we've got a continuous outer longitudinal layer of muscle here. And here's another evidence of one of these Albach's plexus, with these neurons in between the two layers of muscle there. All right, so that's the large intestine. You can see it also has folds. These are also non-permanent folds. They're not known as rugae. They're just folds of the colon, colonic folds, and they're non-permanent. They can expand to enable the passing of feces through the large intestine as well. And again, they contain both the mucosa and then the submucosa. Again, you can see some more lymphoid tissue in this region. This one's a bit slow to load. It's taking its time. It's Friday afternoon, I understand how it feels. All right, so what we're looking at here is a section of the appendix. Now, the appendix is also considered part of the large intestine because it has the same structure. And again, we can see that here. We've got this simple straight tubular gland with some goblet cells here. But, of course, the appendix is another region of the intestinal tract that has permanent aggregations of lymphoid tissue, and it's very disruptive, a bit like what we saw with the Peyer's patches. It disrupts so much that some of the glands aren't even apparent. Sometimes the epithelium can even be disrupted here. So we've got these big accumulations of follicles here. There's a nice germinal center here. See this sort of paler region here? It means these cells have been activated in this tissue. But again, we've got lots of goblet cells, simple straight tubular glands. I've got our muscularis, submucosa here, muscularis there, and obviously a nice serosa on the outside. A bit difficult to see the squamous cells, nuclei there, but it certainly looks like it. Are Peyer's patches only found in the ileum? Yes. It's funny, I was at an anatomy conference earlier in the year, and some people talked about Peyer's patches in the jejunum, but I think those are non-permanent aggregations, whereas the permanent aggregations are referred to as the Peyer's patches, and they're specifically localized to the ileum. Thank you for the question, Ruby. All right, so that's the appendix, and that's also the appendix. So we're looking at, again, even this one's even further disrupted. You can see almost very little evidence of the surface epithelium, the glands, very, very disrupted here. Lots and lots of this lymphoid tissue. Yeah, really not a lot of evidence of the glands. It'll be very difficult to actually identify this as the colon because of how disrupted it is. But if you ever see anything like this, then it's definitely the appendix. This is one of the demonstration slides that we use. Yep. All right, Isabella has asked, could you explain the difference between intercalated, interlobular and striated ducts and where these ducts are located? Sorry, unrelated. No worries. So what's the best way to do this? Let's pop this on. All right, so we will, if we've got time, we'll get back to the ano-rectal junction. We'll finish off our look there. So bear with me. What I'm going to try to do is do some drawing. I think it's a bit easier to explain what's going on here. So let's use a different colour. We'll just start with black for first. All right, so we know that for the salivary glands, they're often arranged into these round sort of structures here. Okay, so we have our salivary glands. Now, these can be either mucus secreting or they can be enzyme secreting, so serous or mucus. And sometimes they can be side by side, and then sometimes during processing, the serous components get pushed out onto the surface, and they can often form these structures called serous demilunes, like this. So that's the serous demilune. Let's say this is mucus, and this is serous here. So there's a serous, mucus, serous, and so on. So they continue, and the first part of the ducts that drains their secretions are the intercalated ducts. Okay, they start out as a simple squamous, and then quickly become a simple cuboidal. So this is intercalated. Intercalated duct. So then that continues, and the cells actually get taller and taller. And the cells, oops, that's a really ugly nucleus, look a little bit like this, and they become known as the striated ducts. Now, if we were to sort of zoom in here, what we'd actually see, that's really bad, is that these cells have these basolateral infoldings, and scattered between them are mitochondria. This is really bad, I apologise. So the cells can have a few microvilli on the surface. That's really ugly, isn't it? Let's get rid of that. Sorry, I'm using a drawing tablet here, and it's the first time I've used it, so I'm getting used to how to use it. Might make that a bit smaller. So let's draw it from the base. So here's our cell. It's got its nucleus there, and it has these intramembranous infoldings. So because it can't have microvilli like on the surface, it's sitting on a basement membrane here. It can have these intramembranous infoldings to increase surface area, to allow it to have more membranes. And in between we can have microvilli. Okay, and so it actually gives the bottom of the thing a striated appearance because the membranes are acidophilic, the mitochondria are acidophilic, and it can actually move substances against the concentration gradient back into the bloodstream. So it can absorb things from the saliva that's being secreted and then move them into the underlying bloodstream here. There against the concentration gradient, and that accounts for the striations. So from the striated duct, they then move into the larger ducts that start out just initially as columnar, and then they may be stratified cuboidal, stratified columnar in appearance. So that's, this would be, this is too thick, interlobular, and then that would go to lobar, and so on. That's really messy and ugly, and I apologize. So if we go back to our slides, let me go back here. Let me find a slide and I'll point those out to you. Right, let's go down. Oops, gone too far. Yeah, we'll have a look at this one. Yeah, so that arrangement to go from intercalated to striated is only found in the salivary glands. There's no striated ducts in the pancreas. What we'll see is that the intercalated and striated ducts are only intralobular, so within the lobules. So it's often difficult to see the intercalated, but these striated ducts are very clear here. So these are the striated ducts. You can see the little, hopefully see the little stripes down the bottom there. There's some nice serous demilunes. And here's our striated ducts, lovely striations here actually. So that's those, and then if we sort of zoom out, the interlobular ducts are found in the connective tissue between. See how this is in connective tissue now? So these are out in the connective tissue. And then if we, zoom out a little bit more, we can then see some of these other ducts here in this connective tissue. So these are the larger interlobular, and then they become the larger lobar ducts. I think we've got some of those there as well. So you can see the sort of stratified epithelium here compared to what we were looking at before. You can see the multiple layers of cells in this bigger gland. So only the intralobular ducts, the striated, the intercalated, are actually able to modify the secretions, whereas the interlobular and the lobar ducts don't modify the secretion at all. They're just basically conduits. Does that help answer your question, Isabella? I know you have said the drawing was hopefully helpful, but hopefully that helped. Great. So if we go back, we'll finish off our stream with having a look at the ano-rectal junction. Right, so I'm going to flip this around. Everyone hold their stomachs. Right. Okay, so this is a section of the ano-rectal junction. We've got the rectum here on the right-hand side, and we've got the anal canal on the left-hand side. There's, again, a little bit of folding here, so sort of ignore this region here. Just sort of follow the epithelium here. So again, what we're doing is we're moving from, this time, a simple columnar to a stratified squamous non-keratinized. Now, that's not always going to help you distinguish between the cardio-oesophageal junction and the ano-rectal junction, but of course the presence of goblet cells will be one of the major factors to look for. because the cardiac and the stomach regions neither have goblet cells. Lots and lots of goblet cells. Okay, simple straight tubular glands. Looks nothing like remember the cardiac region of the stomach? It was those very sparse glands, the very pale staining cytoplasm of those mucus secreting. These are clearly goblet cells, and again, we've got goblet cells on the surface, whereas remember we had surface mucus cells in the cardiac region of the stomach. So if we zoom along to the left here, we can begin to see some of this stratified squamous non-keratinized epithelium, and here we've got the junction. So we're moving from a simple columnar epithelium to a stratified squamous non-keratinized. The cells here are quite pale staining. The muscularis mucosa, you can see it's very disrupted in this region. It's quite scattered. If we zoom out a little bit, we can also take a look at the muscularis. The other thing, look at all these blood vessels. This is very typical of the rectum and the anus, of course. Some of these blood vessels may go on to form things like hemorrhoids and things like that. So it's a very vascular region compared to the cardio-oesophageal junction. Certainly not the same amount of blood vessels here. And then here's our inner circular layer of smooth muscle. Sorry, I'll just zoom in so you can see it's smooth muscle. So again, clearly smooth muscle, lots of small nuclei, very close together. Whereas if this was skeletal muscle, you'd see, lots of big cells. So if we zoom out again, we've got this thickening region of this inner circular layer. This is forming another true sphincter. This is the internal anal sphincter made up of smooth muscle here. Now, don't be mistaken that these are actually skeletal muscle cells. If we zoom in, we can see that they're made up of lots and lots of cells. So these aren't skeletal muscle fibers, which you'd expect in the external anal sphincter. These are smooth muscle, and this is the internal anal sphincter, which is not under our conscious control and is responsive to the defecation reflex. So here's the outer longitudinal layer here. So the things to look for the junction, stratified squamous non-keratinized simple columnar. Look for goblet cells. If you can see goblet cells, then you must be looking at the ano-rectal junction. The ano-rectal junction also has this thickening of the inner circular layer, which is not present. There's no muscular sphincter at the junction between the cardiac region of the stomach and the esophagus. Very, very vascular, as I mentioned, as well. So those are some of the things to look for when trying to distinguish between the cardio-oesophageal and the anal-rectal junction. Okay, well, that about does it for this stream. Does anyone have any last-minute questions before we close down? What's no external rectal sphincter? No, because it's not, the external anal sphincter is not part of the wall, it's actually external, hence its name. It's not actually part of the section. It's external to the actual sort of typical four layers that we see in the gastrointestinal tract. MJ Green, my pleasure. Thanks very much for coming along. I'm glad you found it useful. I'm actually heading overseas next week, so we'll take a break for a couple of weeks, but then we'll get back into it. We'll look at the liver, gallbladder and pancreas, and I'll start to do some CAM 102 supporting streams as well, and then basically anything else. We'll look at the urinary system by that stage, by the time I'm back as well. Histo 101, thanks very much for coming along and being moderator again. So yeah, thanks very much everyone for coming along. I hope you found it useful. Keep an eye out on social media for announcements about the next streams and when they are and so on. So we hope to see you there. Thanks very much for coming along. Thanks to James Crane for sitting next to me and prodding me and Googling things that I didn't know, giving you definitions and so on. Enjoy your weekend, and we'll see you in the next stream. Thanks everyone.